Okay, today we're gonna present about the cloning the ampicillin resistance gene into E. coli cell.
My name is Nalini Khanonwood.
And my name is Varosha Kihasukcharan.
Let's talk about what is ampicillin. Ampicillin is an antibiotic in a group of penicillin, working by eliminating or inhibiting the growth of bacteria, and is commonly used in laboratory research and clinical setting to treat bacterial infection. However, in today's world, most bacteria can resist the antibiotic due to the ampicillin resistance gene. The ampicillin resistance gene is the gene that encodes for a protein that provides the resistance to the antibiotic ampicillin.
And after test the beta-lactam resistant under ampicillin treatment, they found out that the gene that promotes the conjugative transfer is BLA-CYM2.
Cloning the ampicillin resistant genes into E. coli involves several key steps as the following. The first one, isolation of the ampicillin resistant gene. The ampicillin resistant genes can be obtained from a variety of sources.
In this research, the study was carried out in the semi-rural community located at an altitude of approximately 2,500 meters above sea level, east of capital city of Quito in Ecuador. The fecal sample was collected from children and animals such as guinea pig and chicken. And the sample were transported in a cooler approximately 4 Celsius to the laboratory and were processed within 8 hours of collection. In total, DNA was extracted using the DNA Easy blood and tissue kit from Germany.
Next one, preparation of cloning vector. Choose a cloning vector, usually a plasmid from the target bacteria that contain the necessary elements for replication in E. coli. A multiple cloning site or MCS for genes insertion and an antibiotic resistant mark for plasmid selection. It should also have a unique restriction site that match one of the restriction sites in ampicillin-resistant genes.
And in this case, they extract the plasmid DNA using QI PrEP spin mini prep kit from Germany.
And next, the digestion with restriction enzyme. Both the ampicillin-resistant gene and the plasmid vector need to be cut with the same restriction enzyme that generate compatible ends. This ensures that the two DNA fragments will be able to join together. In this case, you should identify a restriction site within the ampicillin resistance genes that match a site in your chosen vector MCS.
Then the next step is ligation, which is mix the digested ampicillin resistant gene and the cut plasmid vector together along with DNA ligase. This enzyme helps to join the two DNA fragments together by catalyzing the formation of two covalent phosphodiester bonds, creating a recombinant plasmid. And next is transformation. We will introduce the recombinant plasmid into E. coli cells, often done by heat shock or electroporation. Heat shock is using high temperature to make the membrane of E. coli have space for the modified plasmid to go inside the cell. And another one, which is electroporation, is using electric shock to push the plasmid into the cell. And after that, we recover it. And the E. coli cells that take up the plasmid will now carry the ampicillin resistance gene along with the rest of the plasmid.
They were inoculated in 10 ml of trypticase soy broth and grown at 37 Celsius for 18 hours. For evaluation of antimicrobial susceptibility by the Dix diffusion method using Mueller-Hinton agar in E. coli ATTC 25922 was used as the quality control strain, which is the step of selection. To do that, 100 microliters of mixture were inoculated by the spread plate method onto nutrient agar supplemented with the tetraxylin inside the colonies, which is cell with ampicillin resistant gene.
In the verification is the verify the presence of ampicillin resistant gene in the E. coli colonies by performing plasmid extraction, PCR, or other molecular biology techniques. Sequencing can also be used to confirm the correct insertion.
And in this case, we use CPR.
And the last step is maintenance and expansion, which is once you have confirmed the presence of ampicillin-resistant gene in your E. coli cells, these cells can be used for further experiments or cloning projects.
And in this case, they use for analyzing the data.
In the ampicillin-resistant gene allows you to maintain the plasmid in the cells by selecting for ampicillin-resistant colony. To summarize, this process results in E. coli cells that carry the ampicillin resistance gene, allowing you to use ampicillin as a selective agent to maintain plasmids containing this gene. It is important to follow proper safety and ethical guidelines when you're working with genetically modified organisms and potentially antibiotic resistant bacteria. Thank you.